everybody, welcome to the channel. So today I will be extracting DNA from two pieces of paper right here. Without further ado, let's get into it, I guess. Alright, now get a needle and then I will poke a hole in these two 0.5 milliliter tubes just in the bottom of it. So this will act as a makeshift filter to filter out, the large paper pieces. If you would like, but I'm focusing on yield right now, you could add a piece of very fine piece of cotton in the bottom of the tube, and that will really act like a filter, filtering out the large particulates. Alright, the next step is just cutting the piece of paper with the DNA in it, right? And what I'm gonna do later is just cut it into tiny strips and put them in the tube. Now the next step is adding into these little tubes that have the paper in them. So these are the 0.5 milliliter tubes with the holes with the hole in the bottom. I'm going to add about 50 microliters of pre-warmed water at about 65 degrees Celsius. And hopefully that will allow the DNA to elute a bit better. Alright, now that the incubation is done, I need to centrifuge this. I'll use about 12,000 Gs for about 3 minutes. Okay, now that got something, it's time to put it on a gel and see if we have some DNA in there.